What's up, penguins? Today we're going to do 2024 number 6 on translation rate. This has a national average of 1.67. Scientists can quantify the rate of translation as ribosomes move along mRNA from one codon to the next. Using a procedure called ribosome profiling, the scientists measured how long a ribosome remains stationary at each codon of each mRNA. They determined the average translation rate across all codons is 5.2 amino acids per second, but that the average translation rate for specific codons in different mRNA sequences can vary widely. These variations in translation rate are thought to facilitate correct folding of the protein being produced. The rate at which 3 different codons were translated were measured in 100 different mRNAs. The scientists determined the distribution of rate, or number of times each rate was recorded, for each of the 3 codons, GAC, AUU, and UGG. So here we see in figure A, we have GAC. And then here we can see in figure B that we have AUU. And then in C, we have UGG. So now we want to say, okay, well, A, using the data in figure 1, graph A, which we have graph A, identify the rate in MS per codon that was recorded the greatest number of times for the GAC codon. So here we're looking at the number of times the rate was recorded, and we're looking for where was it the greatest. So the greatest is going to be our largest bar that we have in our graph. And so you can see right here that at 150, we have the largest bar. It goes up to about 20, while the next closest one is probably, what's that, 17, 16? So we can say that 150 would be our rate that we would find the greatest number. Now, this is an identified point, which means that you are able to write this as just the answer. So the student just wrote 150 ms per codon, and they got their point. Personally, I tell my students to write this as a complete sentence. So the rate is 150 ms per codon. It doesn't take you that long to make it into a complete sentence, and it ensures you get the point. Part B, using the data in figure 1, graphs B and C, as shown, describe the variation translation rate of the AUG codon compared with that of the UGG codon. So here we see at AUU, we have like a very short variation, like it's not really a lot of variation. There's a very little bit of variation. So it goes from 50 to 450. So we have it kind of a condensed, it seems to be mostly in that 100, 150 area, but it's very condensed in that lower region. Versus if we look at UGG, there's a lot more variation. It goes from 50 all the way to 950. So there's a lot of differences in the amount of time or the rate that it took for this translation to take place. So we could say that there's greater variation in UGG codons than we see in the AUU codons. You could also say the actual numbers. UGG range from 50 to 950, while the AUU range from 50 to 450. So you can kind of see the difference in their ranges. Students said there's more variation in the UGG codon than the AUU codon because the numbers are more spread out. So part C, scientists hypothesize that tRNA molecules that bind to UGG codons are available in lower abundance than tRNA that's bind to AUU codons. Support the scientific hypothesis using the data. So we need to come up with something about this data that helps us to support the claim that there is less UGG available than there is AUU. And as we already talked about, there's more variation in the UGG. It must take a lot of time sometimes for it to find that UGG, which means it must be in a lower abundance. So saying that the translation rate of UGG is slower, the translation of UGG takes longer, or there's more of the UGG codons are translated at a slower rate. So that kind of helps us to see that it must be in a lower abundance. So students said that is true because translation rate of UGG codon took longer in general than AUU codons. So part D. Amino acids can be encoded by multiple codons. In many organisms, certain codons for the same amino acid occur more frequently in an mRNA than do other codons. Based on the data provided, explain why the use of one codon over another for the same amino acid might result in increased levels of protein production from a particular mRNA. So as you remember, there was that wobble. Sometimes we see that there is variation where you can have different codons that code for the same amino acid. So this question is asking us, why would there be one codon selected over another? Like, so why would we keep seeing the same codon in a section, even though there's different codons for that same amino acid? And the reason it has to do with that some codons are translated faster than others. We saw based on that diagram that those codons had different rates. And so because some of them are translated faster, that allows them to make more of this protein in a shorter amount of time. So the use of one codon might be faster to translate than another codon, causing increased rate of protein production. So I hope that this was helpful. Remember that if you have penguins, just says bye, y'all.